Hi everyone. Today we're examining a study by Dudko and colleagues from the journal Aging Cell that puts a long-standing tenet of reproductive biology to the test. The research looks into whether the decline in oocyte quality, which is caused by age-related DNA damage and has long been considered irreversible, can in fact be turned back. So let's break down their findings.
For decades, the scientific consensus has been quite clear. As female mammals age, their oocytes accumulate DNA damage, which leads to a steady decline in quality. You see, this process has always been viewed as a fundamental one-way street of reproductive aging. It contributes to lower fertility and a higher chance of embryos with abnormal chromosome numbers, a condition called aneuploidy. This central idea has framed oocyte aging as a simple matter of inevitable decay.
And this brings us to the central question Dudko and his team decided to investigate. Are the molecular signs of oocyte aging truly permanent? Their entire study was designed to find out if the nucleus of an aged oocyte with all of its accumulated DNA damage could be molecularly repaired, and repaired so well that its full potential to develop could be restored.
So here's the path we'll take through their research. We'll start by characterizing the problem itself. Then, we'll walk through the experiments that tested for reversibility, uncover the specific repair mechanism they identified, and finally, look at the results, all the way to live birth, and think about what this means for potential oocyte therapies.
Okay, first things first, the researchers needed to establish a quantitative baseline. They had to precisely characterize the key cellular and genetic differences between oocytes from young mice and those from mice of what's called advanced-maternal-age, or AMA. This AMA group serves as the experimental model for reproductive aging.
Now, if you just look at them under a microscope on a purely morphological level, there are no significant differences. At the germinal vesicle stage, and this is the point of meiotic arrest where oocytes spend most of their lives, the oocytes from young and aged mice appear identical. This tells you right away that the main problems linked to aging aren't structural, but are instead happening at the molecular level.
A molecular analysis, however, tells a very different story. This chart here quantifies the presence of something called gamma H2AX foci. Now, gamma H2AX is a form of the H2A histone protein that gathers at sites of DNA double-strand breaks, so you can think of it as a standard marker for this kind of damage. In young oocytes, about 16% show these markers, but in AMA oocytes, that number jumps more than 4 times, to almost 70%. This confirms that despite looking the same, the aged oocytes are carrying a substantial and measurable burden of DNA damage.
So now that they've quantified this damage, the next logical step for the researchers was to design experiments to see if it could be repaired. The core strategy they used was to place an aged nucleus into a young and presumably healthier cellular environment, using nuclear transfer.
The researchers used 2 distinct nuclear transfer methods. The first was a standard transfer. Here the nucleus, the germinal vesicle, from an AMA oocyte was moved into a young oocyte that had its own nucleus removed. The second, more innovative method, called SEGV transfer, added one crucial step. Before removing the young nucleus, its membrane was intentionally ruptured. This released all its soluble contents into the cytoplasm, creating what the researchers called an enriched cytoplast, basically a cell body that's been supplemented with factors from the young nucleus.
The results from these 2 approaches were strikingly different. The standard transfer of an aged nucleus into a young cytoplasm had almost no effect. Damage levels stayed high, around 67%. This finding is quite powerful. It suggests that the young cytoplasm, by itself, isn't enough to repair age-related DNA damage. But in stark contrast, the SEGV transfer, which enriched the cytoplasm with those young nuclear factors, led to a dramatic reduction in damage. The incidence of gamma H2A.X foci dropped all the way down to 16.4%, a level that's statistically indistinguishable from what you see in young control oocytes.
This result really pinpoints the source of the repair activity. It seems to originate from inside the nucleus of the young oocyte, and it gets released into the cytoplasm during that selective enucleation process. So, the next set of experiments was all about isolating precisely which nuclear component was responsible.
The critical difference between the 2 transfer methods is the presence of the young oocyte's karyoplasm. As it's defined here, this is the soluble part of the nucleoplasm. It's the complex mixture of proteins, RNA, and metabolites that aren't physically attached to the chromatin. The working hypothesis then became that these soluble factors are the active agents behind the DNA repair. To test this hypothesis, the researchers ran a control experiment.
They transferred only the nucleolus, which is a large non-membranous structure inside the nucleus, from a young oocyte along with the aged germinal vesicle. The result on the left shows this had no significant reparative effect. Damage remained high. This experiment systematically rules out a specific structured part of the nucleus as the source of repair. And that reinforces the conclusion that the reparative factors are located within the soluble nuclear fraction, the karyoplasm.
All right, so they've identified the karyoplasm as the source of repair. The investigation then turns to the mechanism. The next experiments explored how these soluble factors within the young karyoplasm might actually be repairing the aged chromatin.
One of the main candidate mechanisms was the RECA cohesin pathway. This was a logical place to start, as the age-related loss of this particular cohesin component has been linked to chromosomal instability and damage before. However, the study found no evidence that new RECA was being loaded onto the chromatin of the reconstructed oocytes. This indicates that while RECA loss might play a role in causing the damage in the first place, the repair mechanism being activated by the young karyoplasm is independent of this specific pathway.
So, the investigation then shifted from specific protein pathways to the overall physical structure of the chromatin itself. For this, they used a technique called fluorescence recovery after photobleaching, or FRAP. With this method, you use a laser to bleach a small area of fluorescently tagged proteins in the nucleus. Then you measure how quickly the fluorescence recovers. A faster recovery means higher protein mobility, which acts as a proxy for a more open or decondensed chromatin structure, a state that's generally more accessible to DNA repair machinery.
The FRAP data, which is summarized in this table, reveals a very clear trend. The AMA oocytes show the slowest fluorescence recovery, which points to a more compacted, less accessible chromatin structure. In contrast, the young oocytes and, critically, the repaired SEGV transfer oocytes both show much faster recovery rates. This data suggests a potential mechanism. The young karyoplasm contains factors that remodel the aged chromatin, making it more accessible. This structural loosening likely allows large repair enzyme complexes to physically get to the DNA lesions and fix them.
Now, molecular repairs and changes in chromatin dynamics are significant indicators, but the definitive test of an oocyte's quality is its ability to support embryonic development all the way to term. So the final phase of the study was to see if these reconstructed oocytes were in fact developmentally competent.
The results of the embryo transfer experiments were definitive. As this chart shows, embryos derived from the AMA control oocytes completely failed to develop to term. This resulted in 0 live births. In sharp contrast, the reconstructed SEGV oocytes, the ones with an aged nucleus repaired and an enriched young cytoplast, developed to term at a rate that was statistically identical to the young control group. This isn't just a partial recovery, it represents a complete functional rescue of developmental competence.
Finally, let's consider the broader implications of these findings. This research directly confronts that long-held view on the irreversibility of oocyte aging, and it opens up new lines of inquiry for both basic science and for potential therapeutic applications.
The author's conclusion is a direct challenge to the established paradigm. They state, our results show that the age-associated decline in oocyte quality can be effectively mitigated, opening new possibilities for cell-based oocyte therapy. This finding really reframes the scientific conversation, moving it away from one of managing an inevitable decline to one of actively exploring molecular reversal.
So, to summarize the key findings, the study demonstrates that age-dependent DNA damage in oocytes is not a terminally determined state. It shows that soluble factors from a young nucleus are capable of repairing this damage, likely through a mechanism that involves chromatin remodeling. And most importantly, it shows that this molecular repair leads to a complete restoration of fertility in their mouse model.
This research provides a powerful proof of concept in a mammalian system. The challenge now is translation. The immediate scientific question is to identify the specific molecules within the karyoplasm that are responsible for the repair. And looking further ahead, the path from this mouse model to any potential human therapy is filled with immense technical, safety, and bioethical considerations that will require careful and systematic investigation.